Hello, and welcome to this webinar titled Analytical Strategies for Comparability in Bioprocess Development, hosted by Biopharma Asia Magazine and presented by Dr. Christine P. Chan and Dr. Joe Barco. My name is Shri Patel, and I will be the moderator. Before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab located at the bottom of your screen, and we'll go through these at the end of the presentation. Now, please allow me to introduce you to our presenters. Dr. Christine P. Chan is currently Principal Scientist in Global Manufacturing Science and Technology Specialty Care Operations at Sanofi. She is a protein biochemist with broad experience in the biopharmaceutical industry, including prior experience at Sandoz Pharmaceuticals and Merck & Co. Incorporated. She specializes in the analysis of recombinant products produced from mammalian cells for vaccines and biologics development. Her application experience includes expression cell line selection and drug product process development, manufacturing tech transfers, and life cycle management of commercialized products. Our second presenter, Dr. Joe Barco, is the Director of Product Marketing at Unchained Labs. He is responsible for new product launches and product development for protein analytical tools. Dr. Barco received his Ph.D. in chemistry from Stanford University. He has spent 15 years in the biotechnology instrumentation area, first as an application scientist, followed by positions of increasing responsibility in product management and marketing. His main interest is bringing better tools to scientific discovery. I would now like to invite our presenter for the first half of this webinar, Dr. Christine P. Chan, to join the webinar. Welcome, Dr. Chan.
Thank you, Shri, for the introduction. I would also like to take this opportunity to thank the organizer, Biopharma Asia Magazine, and the sponsor, Unchained Labs, for this great opportunity today to discuss some details regarding analytical applications for demonstrating comparability after a bioprocess change. A special thank you to everyone who took time out of your busy schedule today to call into this webinar. My first slide shows an outline of my talk. I'd like to start with a brief highlight on the key elements of a comparability assessment, then discuss the background CMC information that each project team would have collected for the specific product during drug development, which forms the foundation that supports the comparability exercise in terms of process understanding, product knowledge, and the assay steps. In the second half of my talk, we'll discuss some specific aspects of planning of the comparability studies, talk a little about the upfront risk assessment piece, then discuss the selection of a set of assays that are appropriate for the comparisons, considerations of the type of stability studies to include, and setting predefined acceptance criteria. Finally, when we collate the data at the end of the study, it is not uncommon to see some differences between the pre- and post-change data sets. So I'd like to cover some perspectives and considerations in terms of explaining and interpreting these differences to see if they may or may not be impacting safety and efficacy for the patients. Bioprocess changes are quite common. At some point, there may be a need for change and improvement anywhere in the process, in the upstream cell culture, downstream purification, formulation processing, as well as in the test methods and associated reference standards. These changes are often implemented during development, but can also happen after licensure. Regardless, we all look towards the key guidance document from ICH, Q5E, issued in 2005, entitled Comparability of Biotechnological Biological Products Subject to Changes in Their Manufacturing Process. And I have extracted here a key quote. The demonstration of comparability does not necessarily mean that the quality attributes of the pre-change and post-change product are identical, but that they are highly similar and that the existing knowledge is sufficiently predictive to ensure that any differences in quality attributes have no adverse impact upon safety and efficacy of the drug product. This next slide shows the key elements of a comparability assessment. Taking a totality-of-evidence approach, we go through 3 key steps of assessment, and at each point evaluate the residual uncertainty of the collective information to see if we need to proceed to the next step. The first step at the ground level is analytical characterization, which includes biochemical, higher-order structure analysis, stability and degradation profiling, in vitro biological activity or potency. The outcome of this evaluation may be a conclusion that the materials are highly similar, or different, and need to be evaluated on a case-by-case basis. If the uncertainty level is high, we then proceed to the next level for additional studies, such as non-clinical studies in animals and clinical studies in humans. Overall, we take a totality of evidence approach to have a holistic overview of the data. Here I have inserted a slide to briefly clarify the difference of comparability studies done as part of a traditional drug development, which is what I'm covering today, and that for biosimilar development. On the left-hand side, the kind of overview, key points of traditional drug development as it relates to comparability studies. The investment of resources in traditional drug development on analytical characterization is a small part, much less than that for non-clinical studies and human clinical trials. There's an expectation that when we move forward in the stages of development, that the product qualities are adequately similar, that we can use the data from the last trial to support the next trial. And then post-approval, with the goal of continuous improvement, we use comparability protocols to demonstrate no impact of the process change. In biosimilar development, shown on the right-hand side, the development focus is sort of flipped. The analytical characterization is the major component. Because the product and process information from the innovator company remains proprietary and is not available to the biosimilar developer. So this knowledge gap dictates that the work starts from analysis of the final product and then work backwards. As such, there is heavy investment on detailed analytical characterization with the hope that there will be less investment on non-clinical and clinical studies. In both cases, the characterization, the analytical characterization, is the foundation of the comparability exercise. There may be differences in the level of details in the analytical work, but the technologies and the data considerations are similar. Some drill down on the background CMC information. It is important that we establish adequate process understanding. We need to define what are the typical ranges of process variations, find out how does process parameter impact product characteristics, and design a solid control strategy for operations. We also need to have sound product knowledge, understand what are the mechanisms of action for drug efficacy, establish some linkage of product attributes to safety and efficacy in patients, what are the structural features that confer function, and certainly define stability profile of the molecule. For the assays that are used to monitor and characterize the products as well as the process, we need to define the capability of the methods in terms of sensitivity, specificity, as well as what type of sample matrix may be applicable. It is also important to define the ability of the assays to discriminate between product variants. And since we often use orthogonal assays to complement each other, we should also know how the assay readout from one assay relates to another. This slide shows a general schematic of the production process of a drug to storage and delivery to the patient. It serves as a reminder that biologic processing is very complex, and the product protein is often subjected to different environmental stress in this whole process. The product quality can be sensitive to particular process conditions, say low pH elution of a protein A column, as well as changes from one condition to another, such as concentration changes and pH shifts in chromatography steps. So there may be chemical instability with covalent modifications and physical instability from conformational, colloidal, and interfacial mechanisms, resulting in higher order structural changes and aggregation. In multi-domain proteins, these instabilities are often interrelated. It is well recognized that proteins are only marginally stable, with the energy barriers between folded and partially unfolded states being small. So because of the increased risk of immunogenicity of aggregation and particulate formation, it is very important to have a well-designed control strategy based on product and process understanding for robust manufacturing. Next slide, talk about product profile knowledge. As we all know, biologic products usually have a significant level of molecular heterogeneity. For product quality attributes that are critical to safety and efficacy, that is the CQAs, it is important to control them within an appropriate range to ensure a desired level of drug quality. For monoclonal antibodies, it is clear that certain PTMs, or post-translational modifications, may affect function, stability, clearance in the body, elicit unwanted immunogenicity, which could impact safety. So it is important to sort this out for each map in development. So I have highlighted some potential modification here in the diagram on the right. N-terminal cyclization, oxidation, deamidation, fragmentation may occur at the hinge region, variation in glycosylated form at the consensus asparagine site within CH2 domain, variation at the C-terminal and lysine processing, and then disulfide rearrangement. These modifications can occur in the cell during production culture and also afterwards in the harvest supernatants and during subsequent processing. And as we have discussed, we need to be mindful of the chemical pathways as well as the physical pathway of potential degradation. In terms of assays, it is important to have orthogonal methods, use more than one method to characterize a given product attribute. In most places, the analytical lab operate a set of platform assays and then case by case for each product, because there is a different set of attributes that are critical for function and stability, would choose an assay set that is specific and relevant for characterization of that particular protein. In terms of methodologies, there are many options to choose from, generally falls within these three categories. The biochemical methods, HPLC, electrophoresis, LCMS, with enzymatic digestion. The biophysical methods, here I've separated them into two subgroups. The methods that shows the bulk properties, there are various spectroscopic methods and differential scanning calorimetry. These assay readout provides ensemble average on the structural information of the sample, and they may not be sensitive enough to show differences, as shown in the example on the lower left side. And then there are the methods that look specifically for aggregation in particulates, such as size exclusion chromatography in conjunction with light scattering. Some data is shown in the middle panel. And imaging technology is shown some data on the right panel that look at particulate size, morphology, and count. Third category, the potency assay. Depending on the mechanism of action, the drug molecule may bind to a biological target, have intrinsic enzyme activity, and these can be measured in a solution-based assay and further verified in a cell-based assay. So overall, the analytical assessment categories that we need to cover in a comparability exercise is summarized on this slide. Again, the results we look to provide should be a comprehensive profile with complementary and confirmatory data. So very quickly, categories include primary structure, higher order structure, product-related substance and impurity, biological function, which we just discussed. In addition, process-related impurity, meaning the impurities from host cells, the residual DNA, residual host cell protein, impurities from the process, such as protein that leaches off the columns, metal ion contamination, residual solvents, and extractable leachables. Properties of the finished drug product, general properties, may include the strength of the drug or concentration, formulation components, and demonstration of sterility. And last but not least, stability data. So we not only need to have real-time, real storage condition data, but also studies from accelerated condition as well as stress testing or force degradation studies. Before moving on to talk specifically about comparability planning, I want to pause here to talk a little bit about defining CQAs and their acceptable ranges. Very often, there is a challenge in interpreting the comparability study results, mainly because we don't have adequate foundation information to interpret them. So it is valuable to plan within the drug development path to test a broad range of variants for stability, bioactivity, and pharmacokinetic impacts. Actually, some of these experiments can be small-scale studies and can be done as part of a process development effort. I've listed here some approaches for expanding the range of product variants. So these additional range of product variants can be generated using various force degradation conditions. They can be versions of the product generated from clone selection studies, process ranging studies, whole time studies. And within just the analytical lab, there can be simple enrichment or fractionation purification of the samples, or use biochemistry approach using different kinds of enzymes to cleave off C-terminal residues or remodel the glycan chains. And then feed these materials into potency assays to look at variations of the effects in, say, target binding and cellular activity. The overall goal is to establish linkage of the different pieces of information to see how PTMs may affect bioactivity and stability. In the left corner, lower left corner, is an example of methionine oxidation affecting stability of the CH2 domain of the monoclonal, as shown by differential scanning calorimetry. You can see that the red trace, the oxidized form, has shifted to a lower TM. So through these studies, we find out more about how various assays are related, the orthogonality of various assays. Finally, it is of interest to evaluate clearance and change of the molecule in circulation. So at this point, there's been many papers with monoclonal antibody examples, And here I am citing a recent open access paper published in 2017 to show just an example of using affinity capture and LC-MS analysis. One can study differential clearance of the different glycoforms in animals, as shown in the middle panel, and also simple plasma incubation study, data shown on the right panel, to see if there is conversion. So now let's talk about comparability study planning. These studies may be done at different milestones during progression of the program. Again, the CMC changes may include upstream, downstream formulation, and assay. The scope of the study and the amount of data that's needed to support the change depends on the nature of the change and when the proposed change are going to be implemented during the product life cycle. In early phase, there's usually a higher tolerance of allowing differences in the product attribute, with the product knowledge and process understanding still to be developed. So there's a high residual risk. But at this point, the key emphasis of the evaluation is safety, with the expectation that residual risk will be sorted out and decreased with additional clinical trials. On the other end of this path is post-licensure changes. With the high level of product and process knowledge, there is in turn low tolerance of differences in the product attributes. However, the expectation of this assessment is clear in terms of what would be the allowable differences. In this slide, I have highlighted the key step and considerations in planning of a comparability study based on our knowledge on the process and the product. So we start with, on the left-hand side, what the process change is and the rationale. And usually here, there's a flow chart or a table comparing the process steps before and after the change, sort of a side-by-side comparison, plus discussion on the risk-benefit for the patient, and then summary of the historical process data. On the right-hand side, regarding the product, we list the identified CQAs, their risk ranking, the acceptable ranges, and associated analytical methods, and then summary of the product data. Putting these together, we point out the potentially affected product attributes. And then on the process side on the left, we identify the in-process monitoring tools and discuss the control points. And for product testing on the right-hand side, we choose appropriate methods based on the capability of these methods to detect what we consider the potential differences. The testing levels include release methods. Very often, it might be both drug substance and drug product, some in-process testing. Extended characterization, very often done side-by-side. Forced degradation studies, also done side-by-side. And then characterization of impurities. As part of the plan, we also need to determine the number of post-change batches to include and set acceptance criteria based on available clinical lots and historical manufacturer ranges. Along the way, we also need to decide if real-time stability study is needed, and if so, how much data to collect before a regulatory submission. At the end of the study, we address the differences and evaluate the impact. Depending on the data, there are two paths. On the left side, we might be able to confirm that there's lack of adverse impact on safety and efficacy, so no additional study needed. Or there may be some residual risk, and then we consider additional next steps, such as perhaps additional potency assays before going to consider non-clinical and clinical studies. This next slide outlines some CMC changes and the potential risk. At the top of the table are some changes that have low potential impact on product quality. And then all the way towards the bottom, types of process changes that have high potential of changing some quality attributes. The example would be changing a cell line with a new transfection or some complex formulation changes. So in these kind of higher risk cases, there needs to be more complete and extensive analytical characterization. So we don't have a lot of time left to discuss this in particular details, and I'd like to refer you to the references, which are our guidance documents that's listed in the footnote. The next slide summarizes some commonly used analytical methods for protein characterization in a comparability study. The attributes of interest are summarized on the left, and we have discussed them in earlier slides. On the right-hand side are various test methods that are options for considerations. So overall, we need to cover 3 levels of testing. Routine QC control, which needs to be validated assays, extended characterization, where the method needs to be qualified for intended use, and then the various level of stability testing. The key is to maximize the potential for detecting relevant differences in product attributes. So a few comments about setting predetermined acceptance criteria. Generally, comparability acceptance criteria need to be more stringent than the release limit. For post-approval changes, we often use the complete manufacturing history. We often have a lot of data, so there's enough statistical power to set tolerance intervals. And common practice is to set the TI at 95% confidence level with 99% coverage. In some cases, that TI level is actually broader than the release limit, and so we do then narrow that to use the release limit. In early phase, there may not be a lot of data to set meaningful statistical criteria. So when the N is small, the TI limit would actually be very wide, which is not appropriate. So in this case, we don't use statistics and just simply report the min-max range with calculation of the mean. In terms of the level of statistics to use, FDA scientists have proposed for biosimilar assessment a tiered approach based on the criticality ranking of the CQAs. So in essence, the more critical the CQA, the more rigorous the statistical comparison. So the recommendation is that for CQAs that may impact clinical outcome, one should consider equivalence testing. For CQAs that had a moderate and low risk, suggestion is to do a calculation of the quality range using mean and standard deviations. And then there's also non-statistical assessment, just looking at raw data such as a gel image or graphical display of spectra. In a comparability exercise, forced degradation studies are often required because they provide insight into potential differences in stability profile and purity and impurity profile. The idea is that small differences may be amplified and detectable under high stress conditions over a short period of time. For these kind of data, we can have a profile comparison. And I've shown a example on the left-hand side, overlay of chromatograms. 3 lots of data from the original process, 3 sets of data from the new process, and a reference control, total of 7 samples. And then there's also situations where you may consider using rate comparisons. So those are the situations where you decide to invest resources to run experiment with multiple time points for kinetic comparisons. The example I chosen here, I wanted to illustrate the point that even for the same process, and that is your pre-change process, there may be variation among the slopes across different lots. And so when we set equivalence acceptance criteria, it's very important to take into account the variability about the slope of the lots of the original process. And then also fold in consideration of the scientific knowledge of the impact of degradation over time. It is clear that with various testing done with all the high-resolution methods, it is not unusual to observe some analytical differences. So the challenge is that structural characteristics of proteins as they relate to clinical outcomes may not be fully explored or understood for a given product at a given juncture of that drug development process. The key question we always ask is then whether the difference is of practical significance. Would the analytical differences be discernible physiologically in the clinical setting? So to navigate the narrative in the report, we often consider the following. If the attribute that fail acceptance criteria would remain within the range of historical manufacturing experience. So in those cases, after appropriate investigation risk assessment, that usually would be acceptable. And then if the differences represent an improvement, if there's lower HCP, higher level of a glycoform that confer function, then again, with the appropriate assessment, that usually is considered acceptable. And there are cases that we can demonstrate same routes of degradation, but the kinetics are slightly different. And so we need to explain these minor differences. Perhaps there are sampling issues or assay issues. And then one more, if we find new impurities in product variants, it is often necessary to go ahead and identify and characterize them and try to explain the formation of these entities in the context of the process change so that we can demonstrate process understanding and potentially modify our control strategy. So overall, at the end of the assessment of the impact of observed differences, the evaluation should indicate whether or not confirmatory, non-clinical, or clinical studies are appropriate. Overall, the considerations of comparability studies design circles around the same questions. How much details are needed and how comparable do the product needs to be under the concept of similar but not identical, as long as there is no adverse impact to safety and efficacy. So my last slide, I have included additional references on over here. You can take a look later. And I'm going to hand the control back to Sri now.
Thank you very much, Dr. Chan. So just as a reminder to our listeners, if you have any questions for Dr. Chan or our next speaker, Dr. Barco, then please click on the questions tab and send them through to us. We'll be asking these at the end of the presentation. I would now like to invite the speaker for the second half of this presentation, Dr. Joe Barco, to present an industry perspective titled Limber Up Your Lab with Better Tools for Comparing Biologic. Welcome, Dr. Barco.
Thank you, Sri, and thank you, Christine, for your exciting, interesting presentation. What I'm going to talk about today is how Unchained Labs takes an approach about developing analytic tools that can address comparability exercises. From our perspective, there are four main characteristics of testing that should be done in this area. One is that the test should be direct and straightforward. So where it's going to be performed in different labs and performed at different skill sets, and kind of having a complicated experiment is not going to be of much help. Second is that it should be rapid. The third is that it should be sensitive, so where it can pick up small changes and small differences in the product. And then the fourth is that it should be reproducible, that there should not be essentially instrumentation questions or determinations about how the experiment is run that impact the conclusions from the experiment. We found in the biologic space that there's a lot of suboptimal tools that have been developed, and while a lot of analytical testing methods have been based in two things, either a qualitative assessment of work, because from an academic perspective that was all that was necessary, or they were actually tools developed for small molecules. So as a result, a lot of work done in the biologics area is hindered by having tools that were developed for other purposes. And while they work and while you can get them to work and you can get data out of them, without being designed with biologics in mind, they are always going to be limited and not necessarily give as good of a result. So that's the approach that Unchained Labs takes with our product development and our product improvement. So Unchained Labs, we've been in existence for about 18 months. What we do is we pool tools that are in the market for analytics and workflows and make those tools better and make them specific for biologics. We have an amount of product development that also goes on. So this tool on the left, which is called Uncle, and I'll talk about that as we go, was developed at Unchained Labs. Another tool, Grunt, which is a workflow-based tool, was also developed here. There are two main characteristics. We'll talk about analytics today. The workflow tools, as I mentioned, Grunt here is a buffer exchange platform that's automated. The automation line goes into larger full-scale automation systems. Grunt itself is a benchtop-based buffer exchange, buffer creation tool. In the analytics side, there are three main product lines we have. One is Uncle, which is screening stability. It's a multi-mode detection reader. Hunk is a tool that does delta G measurements, so it has built-in liquid handling and built-in detection to do those experiments. And then this little guy is called Punk, which is a DLS system, so it's a shoebox size DLS. It's about 5 microliter sample in either a chip or a cuvette. I'm not going to talk too much about that today. What I'm going to do is focus on Hunk and Uncle and how they can be used together in terms of developing in work and comparability assessment. So let me tell you a little bit more about Uncle. So Uncle is as I mentioned, is a multi-mode detection reader. It has 3 detection methods: full spectrum fluorescence scan, static light scattering, and dynamic light scattering. There are many. You can pull the raw data out of the instrument, but what we do is we have application software built in so that it automatically analyzes 11 different applications depending on how you've set up the experiment. It's also low sample volume, so it's 9 microliters a sample at a time, and it can run up to 48 samples simultaneously. Now, the 3 detection methods enable multiple applications. These are the 11 that are sort of built into the software. For full spectrum fluorescence, the key applications are thermal melting measurements, so TM measurements. We do a full spectrum measurement looking for tryptophan exposure as a protein is thermally heated. And so we do TM methods, either full spectrum fluorescence, which is just intrinsic fluorescence from tryptophan. We've also added an application to do the traditional DSF method, which is to use a Cipro dye. For dynamic light scattering, we do the main dynamic light scattering measurements, which are sizing and polydispersity measurements. But because we have up to 48 samples at a time that we can do in a small sample volume, we can also take approaches to measure viscosity at low sample volumes, as well as KD. And for static light scattering, our main applications are are TAG, so aggregation onset measurements, and B22 measurements. So with the multimode detection method, what you gain is you gain the ability to not only conserve sample, but also to save in bench space and save in the number of instruments that you have to maintain and save in the number of instruments that people have to be trained on to use. So in comparison with UNCLE, if I were to do DLS, NTM, NTAG at the same time, I need about half a microgram of sample and the bench space of about 25 centimeters square. That's in comparison to doing a DSC method, which is 400 microliters of sample, all of that bench space, a zeta sizer to do TAG, and then a Dynaprobe to do DLS measurements. So with this efficiency, not only am I able to compare things across instrument platforms, I'm able to know that I'm doing the experiment at the same time with the same sample quality. So what I can do, for example, is I can do a thermal melting measurement at the same time as a thermal aggregation measurement. What that gains is that I'm essentially doing all of the scanning for free on the same instrument at the same time on the same sample. With dynamic light scattering incorporated into the instrument, what I can also do is to do a DLS measurement prior to the start of the thermal ramp, and I can do if I wanted to, I can do a dynamic light scattering measurement at the end of the ramp. So at 95 degrees, I expect most of my sample to be aggregated, of course, but what it can do is confirm, or it's an orthogonal measurement to the static light scattering measurement. We've seen on occasion that things sometimes will not show up by aggregation onset, but they will show up by DLS and vice versa. So it's a secondary check to that aggregation onset measurement. Now, what we can do with this, we've made it a point when we've developed UNCLE to focus on simple analysis. So that is to say that there's no subjectivity imparted in terms of analyzing the data. An experiment is run. Once the experiment is finished actually running, it's all analyzed as soon as it's finished running. The instrument knows what type of experiment you set up as part of the setup process, and then analyzes the data right away. So this is a comparison where we're looking at, I think, 12 different samples here. We've had color coding in here so that there's immediately a recognition of where samples fall in terms of a heat map, where you have high, medium, and low thermal melting or thermal aggregation, depending on what you set your parameters to be. So of course, what we can do is we can use this in comparison for traditional methods like TM and TAG. If we're looking at different antibodies in this particular example, what we can do is we can see that if we do multiple replicates, that we have very good precision from the measurement type, even though it's a small sample volume and even though it's a fairly rapid ramping process. And what we can do is compare that with thermal aggregation onset. So that is to say that as we're measuring TM, we get the TAG measurement at the same time, essentially for free. What that gives us is the ability to see if there's a multi-domain protein, for example, which domain unfolding actually leads to aggregation. So the movement of these across multiple samples or different processes can tell us whether or not the sample is comparable from a colloidal stability standpoint. Adding the DLS information gives another piece of information to this in the same instrument. So as I mentioned, you can do the dynamic light scattering prior to starting the thermal ramp, and you can do the measurement at the end of the thermal ramp. Of course, there's nothing that limits you from making the ramp go all the way up to 95 C. You could stop the ramp at 60 degrees and do that comparison as well. But this is a screenshot from the software where we've looked at the same sample at 15 and 95 C. What we're getting is we get a heat map that shows how all of the samples behave relative to one another. We can get individual spectra where we're looking at a sample before and after thermal ramp heating to see kind of where the aggregation pathway, to see if it aggregated into roughly what size it aggregated at. We also get polydispersity information, which is, for those of you who are not familiar with DLS, the width of the peak is some indication of a sample at a particular size, how consistent and how likely that every particle in that sample is that size. So lower polydispersity, then narrower peak, and more uniform the sample. So while I've mentioned this, we talk about this before and after. What we see is sometimes proteins will aggregate into a certain, essentially everything has an aggregation profile, and in some occasions that profile is another peak that is simply shifted to a larger size peak, as we see on the example on the left. And sometimes that example is a very broad peak that is, as the example on the right, where it aggregates to a much larger size. So this aggregation profile is fairly consistent within a protein in a given formulation that could be used as a tracking mechanism to see if that aggregation profile is consistent as well. Of course, there's nothing that requires you to do a thermal ramp prior to doing a DLS measurement. So we use this as a QC check in our laboratories, which is to just simply measure the sample before. And we've seen very straightforward things, which is that at some times you make the DLS measurement, and other times and you're at an expected width of the peak and expected radius of the sample. And other times you measure and the sample is aggregating directly out of storage. So this in and of itself, very quick check to see sample quality prior to doing any other type of bioassay on the sample. So I cannot go into detail in the time limit of how much all of the experiment types on UNCLE. There are application notes on the website that talk about all of the applications on the platform. So there's 11 in one. These are just examples of 6 of those shown here. I'd like to use the rest of my time to talk a little bit about HUNC, which is probably something that people are less familiar with in terms of a measurement for comparability, which the HUNC itself measures delta G. And so delta G in and of itself is a measurement tool to quantify stability and to really refine good conditions. So if conditions appear to be good by, say, thermal melting or other measurements, what delta G helps do is it's a refinement tool, and so it can be used as an orthogonal measurement to other detection methods. We use chemical denaturation to get a delta G value. And there's another part of this, which is delta G trend, which is if we look at the same protein delta G under multiple concentrations, the change in delta G will tell us whether or not the protein is going to aggregate, and more importantly, how it's aggregating. So we can learn whether it's going through a native state or denatured state aggregation, and how likely that protein is to aggregate in comparison to other proteins. Now, oftentimes I throw delta G up on a slide, and people start kind of scrambling their brains to think about thermodynamics lessons that are long ago buried in there from college. So I'm not going to require any recollection of this other than what delta G is. And so delta G is RT ln K, and K is an equilibrium constant, which is the equilibrium between native and unfolded protein. So delta G can be determined and extrapolated back to room temperature equilibrium measurements. So you can imagine if you have a chemical denaturation experiment, I have 2 antibodies in solution. I'm going to go through an unfolding process as I increase the amount of guanidine or urea. Proteins that take more urea or guanidine to unfold are more stable, and so then I'm going to get a delta G measurement from that. That equilibrium measurement, importantly, of native versus denatured, is also going to give us a quantitative value. So I can do this experiment, and what I get out of that is a delta G. And remember, delta G is going to be a ratio between folded and unfolded protein, so I can actually quantify the amount of unfolded protein at room temperature at equilibrium, which is something that I cannot do by other measurements. So I expect, of course, the higher my delta G, the less denatured protein I have, the more stable I expect the protein to be. So delta G in chemical denaturation is this extremely valuable technique because there's not very many methods to get quantitative information on the amount of unfolded protein you have at equilibrium, so why isn't it done? The reason it's not done is because it's kind of a challenging experiment to set up. So this denaturation curve I showed earlier is normally at least a 24-point curve. If you're talking about a multiple domain protein, as most of us are, it's oftentimes a 32 or even 48-point curve. So having someone at their bench make a 48-point curve with very small changes in urea concentration is kind of a recipe for disaster. So Hunk kind of takes the hard part out of this process and automates the hardest parts, which are the actual generation of the curve. So there's liquid handling built in. So all someone has to do is take their protein stock, make their formulations and denaturants, put that into the instrument. The Hunk uses liquid handling, makes the curves, and incubates them, and then reads them at the appropriate time. So there's a fluorescence reader built in. Again, we're using intrinsic fluorescence to figure out the need for, essentially to figure out the curve. The observable is fluorescence, and then we use that observable to go back and calculate a delta G from that. So now that I've kind of talked about how the Hunk works, we talk about how you'd use this. So oftentimes what we do, and this is an example where it's the same protein in slightly different formulations. So if I were to rely only on thermal stability measurements, at this point, once something is, especially later in the process when something is engineered and formulations are fairly steady and stable, you don't see a whole lot of change by TM at that point. So this example is very small changes in formulation, the same protein, TMs are essentially identical. So if I were to look at these 6 samples, they would all look exactly the same to me by TM, and I'd need some way to confirm that these are actually the same. And so delta G is a good orthogonal measurement to that. So if I take these same samples and the same 6 formulations, and I run them on the Hunk, what I can see is that there's large differences in some of these samples in comparison with the delta G measurement. So right away I can see there's something wrong or different with sample 2, where now the amount of denatured protein I have in equilibrium is between 60 and 300 times more than the other proteins. So this orthogonal measurement allows me to identify that something is wrong or different in that sample, and I can investigate the process that created that sample, where otherwise with just the TM measurement or just this other measurement, I would not have had anything to go on in terms of identifying which protein is better or not equivalent to the others. So that's talking about colloidal stability. As I mentioned earlier, aggregation is also something that you can get through delta G measurements. So delta G trend is essentially how we call the term where we're essentially measuring delta G, but changing the protein concentration. So if there is no aggregation that's going to occur, delta G should be concentration independent. So I will see, as I see it shown in the green line, no aggregation. At any concentration of protein I run, I'll have the same delta G. If my protein is going to aggregate, what that is doing is driving equilibrium towards, if it's going to a native state aggregation, which is folded protein aggregating, equilibrium is essentially driven to folded protein. And so in that equation for equilibrium, I will see an increase in delta G. If my protein is aggregating through a denatured state, I will drive equilibrium towards unfolded protein, and my delta G will decrease. There is a paper reference here. I unfortunately neglected to put it on the slide itself, but I can be happy to forward that reference to other people to review. Now, how do I use delta G trend? So oftentimes when we screen things like TM and even delta G, we'll look at one concentration just from a simplicity standpoint. So how this delta G works is if I happen to do this on a protein, I will see different delta Gs at a certain point. So let me take an example, which is Herceptin. Now, Herceptin, the commercial formulation has trehalose and tween in it. Obviously, these are not added simply for fun. They are added because they are to prevent something going on. So if I took the trehalose out of the commercial formulation, or if I took tween out of the commercial formulation, I see that that has an impact in delta G. That delta G measurement actually goes up. So what that's saying is that the protein in a formulation without trehalose and tween is more stable at this concentration than the commercial formulation. So while this is reversing back out of the Herceptin formulation, I know that trehalose and tween are often used to minimize aggregation. So I should be able to see aggregation. I should be able to see that if I had run a delta G experiment. So what I'm going to do is essentially back out of this Perceptin formulation and then run a delta G trend in these different formulations to sort of check that. So here is my screening concentration. That's the light green line. And in that concentration, I can see that the delta G for the minus trehalose and minus tween formulations are actually higher than the commercial formulation. But the distinction here is that if I were to run at multiple concentrations, if I did not have those additives present, I would see delta G decrease. And that's telling me that I have denatured state aggregation occurring with that. At the same time, the commercial formulation, the blue line, is consistent across. All delta G is consistent across all concentrations, which is telling me that I do not have aggregation occurring. So clearly, this technique was not necessarily used for Herceptin formulation development. I cannot say that one way or the other. But what I can see is that within a one-day experiment, I could have identified that the formulations without tween and without trehalose would have led to aggregation. So in comparison with the other measurements I'm getting, so delta G, the lower, at the concentration I tested, delta G with the commercial formulation was lowest and higher without the additives present. But if I started to look at aggregation pathway, and if I use that data in conjunction with the delta G, I would see that the commercial formulation had no aggregation occurring, while both of the other applications did have it occurring. So delta G measurements in and of themselves at one concentration will tell us about stability. Delta G trend, this is delta G at multiple protein concentrations, will tell us about aggregation. So using that data in conjunction with other experiments can give some indication about not only stability, but also aggregation propensity in the aggregation pathway. So in the short time that we've talked today, I hope I've given you some indication that with better tools and the right approach to doing assays on stability and aggregation, that you can get a more elegant result. And with that, I would like to return the controls over to Sri. Thank you.
Thank you very much, Dr. Barco. So at this point, I'd like to begin the question and answer session. So the first question, I believe, is directed for Dr. Chan. You mentioned forced degradation studies. What are some of the recommended study conditions for protein biologics?
So for forced studies for proteins, there are certain key ones that we usually do. So obvious ones are the temperature stress, and very often our drug product would be at refrigeration temperature, and so our temperature stress would be, say, 25 and 40, and it could be higher if it's a very stable protein. And then a light exposure, chemical oxidation, and then mechanical stress. And so these could be done early phase as part of your formulation development or your developability assessment of a product candidate. And then based on that, when the product gets developed, then if there's nothing happening, say in a mechanical stress situation, then you may not have to carry those things forward in a later comparability study.
Great. The next question, I believe, is for Joe, because it's talking about hunk. What is the day-to-day reproducibility on the hunk?
So day-to-day, what we see is we've done this experiment where we've looked at essentially the same sample 12 different days, 12 independent runs, and the delta G is within 0.1 kilocalories. So the reproducibility of the liquid handling part is within 5% precision and accuracy, and then the actual measurement you get out of that is 10.1 kilocalories. So that's only a couple percent difference.
Great. And the next one, I believe, is for yourself as well, Joe. Why are there differences between 2G's values in the commercial formulations?
So the delta G values for Herceptin, so the protein will unfold in distinct domains. So the Fc portion will unfold, and the essentially variable region will unfold separately. So you get one delta G from one part unfolding, and then as you add more denaturin, you get a second jump in delta G.
Great. Next one for Dr. Chen. You mentioned that there are changes even after a drug received licensure. What are some of the changes that are necessary after licensure?
So there could be a variety of reasons. A lot of times at licensure, we would then before licensure, we may plan for a additional manufacturing site. And so post-licensure, we may then set up a new manufacturing site in a different continent. And then there are actually also situations where with the continuous process improvement initiatives that we want to have sort of more environmental control and in terms of prevention of adventitious contamination and procedural things that could also be part of those changes, as well as changing primary reference standards. And then for legacy process, there are still processes that are in serum. So there's a risk-benefit situation for the patient that we do go from a serum-containing process to a serum-free process.
Great. Next one's for Dr. Barco. How does the delta G trend in microgram per mil of protein concentration, shown in your slides, correlate in a real case scenario for an antibody solution at 100 milligrams per mil and proportion of aggregation? I hope that makes sense.
So if I can restate the question is, how can you take a measurement of protein at 100 micrograms per mil and correlate that to something that's going to be happening at 100 mg per mil? So, in this scenario, if you had a 100 mg per mil protein solution and try to measure delta G, and if you're looking for aggregation, it has probably already occurred. So kind of the business end of this is when you're at a low concentration of protein, and you can see small changes and extrapolate that back to what you have theoretically as a protein concentration. So ultimately, what it comes down to is if I were to do the same measurement, if I had a flat line at 100 micrograms per mil, I'd see a flat line at 100 mg per mil. You can kind of see on that curve that I showed a few slides ago, the red line is basically kind of tapering off, and essentially aggregation has already occurred. That's what we do at low concentration.
Great. The next question is for Dr. Chan. You talked briefly about bioassays. Do you have to include bioassays in the comparability study?
So I guess short answer is depends on the program. So usually cell-based assays are not sensitive enough to tell minor differences or adequate enough to explain differences. However, a lot of us still use cell-based assay as kind of a catch-all type of verification to substantiate that there are no unexpected differences from our comparability studies.
Great. The next one's for Dr. Barco. What advantage does full spectrum fluorescence on UNCLE have over just measuring fluorescent ratio like DSF?
I think kind of the way I was saying, and Christine kind of alluded to this, is every protein and every process is different. And so if you were to rely on a ratio of 330 to 350, some proteins just don't have that redshift occurring the same way. In some cases, certain formulations just don't allow it to change. So a full spectrum data gives you options to analyze the data essentially in different ways. So what we will do is we use what's called variation most of the time, which is to look at essentially with how that peak width point time. So you just get more data to work with in comparison to just the ratio.
Great. The next one's for, I think this one's for Dr. Chan. Can you comment on the regulatory requirements for characterization for MAA and IMPD, and are there differences?
So let's sort of discuss a little bit investigational medicinal product dossier. So that's early phase versus marketing authorization applications. So there is a difference in the sense that in the early submissions, the emphasis would be on safety. So the characterization then tends to focus on product and process-related impurities. So the key point is that if you have a higher level of, say, residual host cell protein in your clinical lot than your tox lot, so that makes it challenging to have that developmental continuity. And so the other example would be if you have a higher rate of degradation, higher aggregation material, so that would be a flag for that safety-focused assessment. And so there are differences, but I do want to also mention is that beyond just what stage you're in and you need to be phase appropriate, a lot of you probably already recognize is that across the globe, different agencies had different expectations. So they may not be completely aligned even at the same stage, depending on the health authority either.
Great. The next question, not sure who it's directed at. How do you address the differences between processes when it passes the specification but fails the comparability specification? What are the tests or studies we can do to justify the process improvements?
So I think that's for me. So again, it depends on, right? It depends on what that attribute is, and hopefully there's a sound narrative to justify. Yes, we recognize that this failed what we think are comparability criteria, but there's clinical experience, and so we just need to build a narrative to evaluate whether that's okay in terms of no impact on safety and efficacy. So there's definitely, it's not just a narrative, it needs to have additional testing. So the consideration would be, let's say it's a glycoform, then you only have one assay. Then do you want to go additional? I mean, some of us work on glycoproteins with multiple glycosylation sites. And so in those cases, beside the aniline glycan assays, you'll have supplementation of that data with, say, a glycopeptide map and so on. And definitely, to me, from my perspective, additional orthogonal potency assays that are relevant to the mechanism of action, and then try to build that narrative.
Great. The next question. You mentioned cell-based assays have a larger variation, so often not sensitive enough to detect small differences. What would you recommend for cell therapy products where potency assays are mainly cell-based assays and analytical assays are quite difficult to implement?
I have to say that that's outside of my expertise as a protein chemist. So within kind of my world, cell-based assay, when they have more variations, then we try to control the indicator cell lines, and so to that effect, we minimize the variation of the assay per se. And then the other component of that, and that part, I hope that it's applicable to your question, would be try to tease out the mechanism of action and define what is really those cellular signaling pathways that confer efficacy in humans. And so try to find a less variable potency assay that could be then kind of your surrogate assay to give that linkage.
Thank you very much. So that's all the questions that we have today. So I would like to take this opportunity to thank our presenters, Dr. Christine P. Chan and Dr. Joe Barco, for sharing their knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com. Thank you very much, everyone.